think this is working. So in particular, we are interested in the Bamford-Viere kingdom, named like that because of Dennis Bamford, also known as the double jelly roll viruses. As you can see here, these viruses infect all sorts of hosts from bacteria and archaea all the way to animals. And we are interested in understanding how these viruses assemble. And you can see they have double-stranded DNA genomes, and the genome size ranges between 10 kilobase pairs all the way to the megabase pair. And capsid diameters range between 60 nanometers and almost half a micron. So as you can imagine, in this upper range, these viruses are already competing in size and complexity with some of the smallest cells known. Why are they called double jelly roll viruses? Because this is how the major code protein of these viruses folds. It has a double beta barrel, and these two beta barrels are located perpendicular to the capsid surface. So these proteins make trimers, but because they have these two jelly rolls, they have a pseudo-hexagonal shape. And this shape allows these proteins to fill the six-fold coordinated positions in the icosahedral capsids. So we are solving structures of double jelly roll viruses infecting unicellular organisms from bacteria to unicellular algae. But the main player of my story today is here on the other side, is adenovirus. Adenoviruses are also double jelly roll viruses, and they infect animals, including us, humans. As you can see here, adenoviruses are pathogens. For example, they are one of the leading causes of diarrhea in children. But of course, they are also widely used as therapeutic vectors. And here in this picture from the COVID times, you can see that after the mRNA vaccines, the next most used vaccines, they were all based on adenovirus vectors. So we want to understand how the adenovirus particle is assembled in the nucleus of the cell and how it disassembles when it enters a new cell, and how correct assembly is linked to the possibility to do a correct disassembly to infect a new cell. Of course, we use electron microscopy in all its flavors, from plain resin-embedded cell sections all the way to cryo-electron tomography or single particle averaging. But today I want to tell you about our results in combination with another technique, which is atomic force microscopy. And this is not something we do ourselves. This is a long-standing collaboration with a group of Pedro de Pablo at the Department of Physics of the Condensed Matter at the University in Madrid. So what we do with the atomic force microscope is, as you know, this consists of a cantilever that can scan a surface. We have the virus particles attached to a surface, and importantly, they are in liquid, so they are close to physiological conditions. And with this cantilever, we can obtain images of the topography of the virus particle surface, but we are actually touching the virus particle. So we can interact with it, we can press on it, and measure mechanical properties such as stiffness. We can figure out if a virus particle is soft or rigid. And also we can do this sort of experiment that we call mechanical fatigue experiment. What we do here is, with the AFM, we take many images of the same virus particle over and over and over. And for each image, we are using very weak forces. So with just one image, the virus particle is happy, nothing happens. But when we keep imaging it again and again and again, the stress accumulates. And in the end, at the same time, we are inducing and monitoring the disassembly of the particle and the mechanical stress in real time, as you can see here with an adenovirus particle. So this is an adenovirus, non-enveloped icosahedral particle, rather large, about 100 nanometers in diameter. And most of the capsid is composed by this double jelly roll major code protein, which in adenovirus is usually called hexon. At the vertices, we have the penton base and the fiber, and these two proteins are involved in attachment to the receptor. And then in adenoviruses, in all adenoviruses known, we have these minor code proteins that are located on the inner capsid surface. These are called 3A, 6, and 8. 6 is important, and I'm going to be talking about 6 a lot, because it has membrane-lytic activity. So this protein is needed for the virus to rupture the endosome membrane to escape into the cytosol and travel to the nucleus. Then some adenoviruses, including the human adenoviruses, have an external decorating protein called protein 9. There are over 200 reported adenoviruses, and they infect all sorts of vertebrates. And regarding structure, or in general, actually, the most studied adenoviruses are those used as vectors. So in our group, we wondered what makes different adenoviruses different. And to try to answer this question, we started solving structures of other adenoviruses. For example, we solved the structure of enteric human adenoviruses, but also more exotic viruses, such as adenoviruses infecting reptiles. And from these viruses, I want to highlight what we observed about the external decorating protein. This is the decorating protein in human adenoviruses, and this is the decorating protein in the lizard adenovirus. And as you can see, they fold very differently. Actually, in the lizard adenovirus, this decorating protein folds exactly the same as bacteriophage tail spikes, making us think that these reptilian adenoviruses at some point picked up this gene from a bacteria or a bacteriophage, maybe by sharing the same environment in the animal gut. But although these proteins look so different, they actually share a common motif here, what we call the crystalion. This is the capsid binding motif. And by looking at these structures and by comparing the protein sequences, we came to the conclusion that this gene, the decorating protein in the lizard adenovirus, at some point was duplicated, and each copy evolved in a different way, so that one of the copies kept the capsid binding motif but picked up this different domain, and the other copy, remarkably, lost the capsid binding motif and kept this domain. And this is the E1B55K protein. This is a non-structural protein in human adenoviruses that is actually an oncoprotein. It deregulates the cell cycle during infection. Then we also were looking at adenoviruses infecting chicken. And from this adenovirus, I want to highlight this protein. This is one of the internal minor capsid proteins located here in the vertices. It's the yellow protein. This protein is conserved in all adenoviruses. But as you can see, in each of the 3 adenoviruses that we studied, this protein has a different conformation. Even if it has 2 domains and the 2 domains fall in the same way in the 3 viruses, the position of the 3 domains is different, the relative position in each virus. And this is something that so far the structure prediction algorithms are not picking up yet. Anyway, not everything is capsid in adenoviruses. Inside the adenovirus capsid, there's a lot of stuff. We have the genome, 35 kilobase pairs of double-stranded DNA in the human adenoviruses, and this genome is bound to a large amount of virus-encoded positively charged proteins, 5, 7, and mu. You can see here the charges they have, and here the copy numbers. Now, how is this genome and these proteins organized inside the adenovirus particle? We don't know. We know that there is a 12 micron long DNA molecule together with 25 meter-Dalton of protein inside a 100 nanometer diameter shell. So how is this organized? Back in the 80s, there were some virus disruption studies followed by platinum shadowing EM, where they obtained these kind of images suggesting that this genome and these core proteins were organized in a way similar to nucleosomes in the cells. In single particle averaging with icosahedral symmetry imposed, we just don't see anything in the core, just flat, very level, right? Because it doesn't follow icosahedral symmetry. Several years ago, we gave cryo-electron tomography a go to try to see if we could discern anything in the core. And actually, we didn't get much information, but we could do something with these kind of 3D maps from cryo-electron tomography. You can see that everything is very crowded here to try to do subtomodal averaging up to now. But what we did was to pinpoint the center of each one of these dark blobs, and we gave this data set of blob coordinates to our collaborator, Tony Sieber in Zagreb University. Tony Sieber is a theoretical physicist, and he used this data set to simulate a collection of particles inside an ecosahedral cage. And then he calculated some statistical parameters for these particles and compared the experimental parameters in green with models for the same parameters for theoretical models of particles inside a cage. If we consider that the particles are located at random, we can see that the experiment and the theory don't agree very well. But if we consider that these particles are related to each other by a soft electrostatic repulsion, we can see that now the experiment and the theory agree much better. So the conclusion here was that we could model the adenovirus core as a fluid of soft repulsive particles. And this electrostatic repulsion makes sense because we have, if we just count the negative charges from the DNA and the positive charges from the proteins, we figure out that at least half of the DNA negative charges are not screened by the protein positive charges. And the fact that there is electrostatic repulsion inside the capsid is suggesting also that there must be some kind of internal pressure because this DNA, the different regions of the DNA molecule want to come apart. So another interesting aspect of these adenovirus core proteins is that they are cleaved by the maturation protease. In adenovirus, maturation consists on the cleavage by the protease of several capsid and core proteins. This protease is packaged together with the genome, and it uses the genome as a track to travel inside the capsid and reach its over 2,000 targets. When the protease is not active, we get apparently normal particles with everything in there. The genome is packaged. The capsid and core proteins are there in their precursor version, but these particles are not infectious. And it was already known that they are not infectious because in the endosome, they don't encode properly. And so they don't expose this internal minor code protein, protein 6, that I told you about earlier on, that has the membrane-lytic activity. So these particles cannot open the endosome membrane and they become trapped in the endocytic root and the infection is aborted. So we started trying to figure out why is the immature particle unable to encode. And we used several techniques, but today I want just to show you what we saw with the atomic force microscope. When we do this kind of mechanical fatigue experiments, this is the real-time disassembly of an adenovirus particle. And what we can see is that the first thing that happens when it starts again is that the first thing we lose are the vertex proteins. We start seeing holes here, then a crack develops, and then the particle releases its contents and collapses. So because we know how many images we have taken when each one of the pentons is lost, we can estimate the amount of energy that we need for the virus to start losing pentons. And we observed that in the immature particle, which is this mutant, TS1, we need more energy to make it lose the pentons. The pentons are more stable in the immature virus. Also, when we look at the final disassembly product here, and we plot the height of this particle, we see that in the mature particles, the height is very small, and this corresponds to just the lower part of the capsid staying attached to the substrate. However, in the immature particles, we have a larger diameter of the final product, and this is consistent with the presence of not only the capsid, but the core, the genome and proteins still there, with practically the same diameter they had at the beginning. And we also observed this in cryo-electron tomography, observing disassembly intermediates in the mature virus particle after disruption. We see capsid fragments and the genome is practically gone and it spills every which way. However, in the immature particles, the capsid fragments are there, but they are still attached to the core and the core is still a pretty compact sphere. So this is telling us that during maturation, the adenovirus core is becoming decondensed, it's losing condensation. So how does core condensation then relate to the lack of uncoating and the lack of penton release, or the stability of the pentons? And the answer to this question came again from the AFM. And remember that I told you that we think there is some kind of internal pressure inside the adenovirus particles, so we thought the internal pressure might be destabilizing the pentons. But we needed to actually have some more evidence confirming this internal pressure. So what we did was to measure the stiffness of different adenovirus particles by touching them with the AFM, but without breaking them. And we measured empty capsids, no genome. There's a certain stiffness. Now, immature capsids with the genome inside, they are stiffer. This makes sense because now there's something inside. Now, mature particles, they are even stiffer. So this was suggesting that, yes, there is some internal pressure, and the internal pressure grows during maturation. But the question here was, okay, is this stiffness due to internal pressure or to having something inside which also becomes stiffer? So we needed to measure the stiffness of the core. And to do this, we kept pressing on the virus particle until it breaks open, and the cantilever is, the tip of the cantilever is actually touching the core, and we measure the stiffness of the core. And we see that when the particle matures, the capsid from outside becomes stiffer, but the core becomes softer. So the core decondenses again. And then the next question is, okay, this increase in stiffness is not because the core becomes stiffer. So it agrees with the hypothesis of internal pressure. So is this internal pressure related with the electrostatic repulsion of the DNA? To answer this, we incubated the particles with a DNA condensing agent, spermidine. So if we incubate the empties with spermidine, nothing happens. If we incubate the mature virion with spermidine, again, we recover the mechanical phenotype of the immature particle. So what we conclude from here is that the cleavage of the core proteins during maturation is somehow decreasing this condensing action of the core proteins and increasing the internal pressure of the particle. So this is our model for how maturation relates to uncoating in adenovirus. We have the genome and core proteins. Now the protease cleaves these core proteins, and the condensation action is now decreased. So electrostatic repulsion kicks in, and this exerts pressure on the inner walls of the capsid. And this pressure destabilizes the pentons, which are by definition the weakest part of an icosahedral particle. So when this particle is now metastable. So when it receives the appropriate signal, such as binding to the receptor or acidification in the endosome, these pentons are released. And this opens the way for the exposure of the lytic peptides, allowing it to break open the endosome membrane. So after these results, we started wondering, okay, so we have 3 core proteins. What's the specific role of each one? And we started looking at particles without each one of the core proteins. And first we looked at particles without protein 7, which is the most abundant of the core proteins. And the first surprise came from the paper by our collaborators here that showed that actually you can remove this protein and the genome is still packaged. So wait a minute. I just told you that we need these proteins to condense the genome so that it can fit inside the particle. So what's going on here? What is actually the role of this protein? We looked at cryo-EM reconstructions, and the core organization, well, as you can see here, if we really force the contrast, we start seeing some kind of concentric shells, similar to what we see in viruses packaging naked DNA. But we don't see much really. What do we see with the AFM? What we see is that this is a kind of experiment that we call multiple nanoindentation. In this experiment, we press on one particle until it breaks open, we release it, and then press again until it breaks a little bit more, and so on and so on. And what we see is that at some point, well, we see the crater here when we pressed, and some yellow stuff appears on the background. This is the DNA. This is the genome being released from the virus particles. So when the core protein 7 is not there, this genome is released much earlier. Just after the first nanoindentation, this genome is already out. So when 7 is not there, the genome release is facilitated. Then we again looked at the internal pressure of these particles. Particles without protein 7 are the stiffest, and when we add spermidine, they decrease their stiffness, so we are decreasing the internal pressure. And the same thing with the core. When 7 is not there, we can reach with the FM tip much deeper in the core, telling us again that this core is less condensed. So this is telling us that yes, protein 7 is condensing the DNA, and it's actually modulating genome release and internal capsid pressure. So, but still, it's not needed to package the genome. But what is it needed for? So now I can just remove this censorship here, so to allow you to see the second part of the title. And the second part is, okay, yeah, they can package the DNA, but they are not infectious. So what is 7 doing here? Our collaborators had already seen that these particles without 7 became trapped in the endosome, same as the immature particles. And it becomes they become trapped in the endosome again because they cannot release, they cannot expose protein 6. When they did this experiment where you label protein 6 with antibodies, which can reach it only when it is exposed outside of the capsid, in a wild-type infection, you can see that the label for 6 increases during the first stages of infection. However, when 7 is not there, this increase doesn't happen. So when 7 is not there, protein 6 is not exposed. Okay, so everything is the same as in the immature particles, so we thought, okay, so maybe these particles without 7 are also not releasing pentons, and it's just the same. So we look at penton release with the AFM, and we found the opposite. Actually, when 7 is not there, the pentons are released much, much faster. So there is a way for protein 6 to come out. Why doesn't it come out? So we started looking at the structure. What do we know about the structure of protein 7? Very little, actually. What do we know about the structure of protein 6? Well, a little bit more, but not much. And this is known from the work of Hong Xiao. And what I show here is what they could trace. This is a hexon, and this hexon has a cavity. Here is a hexon trimer. There's a cavity here, so the genome would be down here. And this is what they could observe. They observed an amino terminal peptide of protein 6, some part of the central region of 6. These are the cleavage sites for the maturation protease. And they observed this yellow peptide here, which is one of the peptides cleaved from protein 7. And the interesting thing here, and the surprise, is that this protein 7, which is a DNA binding protein and nobody knew it had anything to do with the capsid, is here binding the capsid, binding hexon. And it is actually binding to the same binding site as protein 6, because remember, this is a trimer. We have 3 binding sites here. One of them is occupied by protein 7. And the position in which these peptides are located here causes that the cleavage site is hidden because the adenovirus protease, this is the crystal structure of the adenovirus protease, comes traveling along the genome. And when it reaches here, it cannot really see this cleavage site. So something needs to happen for this cleavage site to occur. So we looked at the structure of 7 minus particles in comparison with wild-type particles, and we calculated the difference map. Here in red, this is again 1 hexon, and here in red the difference. And the difference very nicely overlaps with the location of protein 7. The peptide protein 7. This is just 1 hexon. We have 4 hexons in the asymmetric unit. If we look from inside the virus, this is the same difference here. But we see other equivalent differences in the equivalent sites in other hexons. And if we lower the threshold a little bit more, we actually see that we have red blobs in all the hexons. So this confirms that protein 7 can bind to all equivalent hexon sites in the asymmetric unit, and that proteins 6 and 7 must be completed for binding to the same site. Then we have another surprise when we looked at the maps, and it is that when 7 is not there, we do have some extra density inside the hexon cavity. So remember, these particles are lacking 7. So why do they have extra density if they are lacking something? Another thing we knew from our collaborators' work is that in these particles, protein 6, when 7 is not there, protein 6 is only partially mature, meaning that this cleavage doesn't happen. And when we look at this extra density, we see that actually we have this kind of bow of density inside the hexon cavity that seems to connect to the tracing of the amino terminal peptide. So this is consistent with these tubes of density corresponding to the next part of protein 6. Now the interesting bit here is that the next part of protein 6 contains the lytic activity, the membrane lytic activity. So if this part is trapped here inside the hexon cavity, there's no way this virus can escape the endosome. So we could propose a model for the structure of this protein, and what we will see here is that when 7 is not there, protein 6 becomes trapped inside hexon. Because it is trapped, the protease cannot reach its cleavage target. This virus cannot have membrane lytic activity, and so it becomes trapped in the endosome. And this is the model that we propose for this competition of proteins 6 and 7 during assembly, and how this results in the lack of proper uncoating. So we have a hexon here, the two proteins want to bind to the same site, and we have competition here. And one important point here is that we have 720 sites for much more than 720 combined copies of 6 and 7, so they need to compete. So during this competition, some of the molecules are bumped out of the hexon cavity, and now the protease can see them and cleave them, and the virus matures. When 7 is not there, we have more than enough sites for all the copies of protein 6 to become happily hidden inside the hexon cavity. Nothing is pushing them out of the cavity, and so they are trapped there and they don't mature. So what are we up to now? This was a 5-angstrom resolution map. We have now the same particle at much better resolution, 2.8 angstroms. And of course, we want to understand more about the structure of this protein because it's an important protein for the infection. However, even at 2.8 angstroms, localized reconstruction, classification, and so on and so forth, we are better than before. We now see helices here, but as you can see, connectivity is not ideal. This is a very difficult problem because we have to remove or somehow get rid of the icosahedral symmetry in position. We have uncertainty regarding the stoichiometry. We may have 1 or 2 copies per hexon trimer, and there are 3 possible orientations for the protein inside each hexon trimer. And also, it's a very small amount of pixels, very small signal, and it is surrounded by the very strong signal of the hexon trimer. So it's so we will keep banging our head against this wall for a while, I think, before we solve it. But in looking at this protein 6 density, we found something else that we had never seen before, and it is that the protein 6 density continues going towards the virus genome. And as you can see here, this is this density inside the hexon cavity, and it has this little leg that connects to the outer layers of the core, where the genome is. And this happens not only in one hexon, but also in other hexons, as you can see here. Here we don't have the hexon, we have removed it. We only have this internal density. But we have these legs in more than one hexon, but not in all hexons. They are in some specific positions. And where are they? If we again look at this forest of densities, if we now place the internal minor code proteins, 3, 8, and 8, these legs are directly related to the internal minor code proteins, and they are actually making a cradle to hold them. So this must be important elements to organize the assembly during the capsid during assembly. Now, the 400 million question, what are these connections made of? We don't know. We suspect they, again, have something to do with this protein 6, but so far we don't understand what's going on here, so we need to keep working on this. What about the other core proteins? So far we have looked at the second one, protein 5. Protein 5, it was known previously that it's located at the periphery of the core, reaching it to the capsid, and it is required for successful genome delivery. When 5 is not there, there are less genomes that arrive at the nucleus. So remember, this is again a positively charged protein, so we expect it to have the same thing as with protein 7. More internal pressure, less core condensation, and so on and so forth. Well, we found exactly the opposite. When protein 5 is not there, the DNA remains much more condensed, and it actually doesn't leave the capsid when we completely disrupt it with the AFM, as you can see here. So, it has the opposite effect as we expected. Our current model for this protein is that because it's bridging capsid with core, when we break the capsid open, capsid fragments are attached to the core through protein 5, so they pull from the DNA, and the DNA can be released. When 5 is not there, this bridge does not exist, so the capsid fragments just fall off, but they don't pull from the gene. So, 2 minutes about genome packaging. Usually in the DNA virus field, when we talk about genome packaging, we have in mind bacteriophage. Bacteriophage assemble pro-capsid, empty, with a molecular motor in one vertex, and this molecular motor pumps the DNA in this anti-capsid. Adenovirus is a double-stranded DNA virus, however, our evidence and evidence from other groups points to this virus actually packaging the genome in a different way, in a concerted way, by growing a capsid around the condensing, the genome being condensed by the core proteins. This is just conventional electron microscopy. So this is our current model for adenovirus assembly. There is this packaging protein that helps to put together capsid with core. The capsid grows around the core. When we have the protease, the whole thing matures. When we don't have the protease, we have the immature particles that I showed you. And then in this process, sometimes things happen, and these particles that were on their way to assembly lose the genome. And we end up with empty shells with different degrees of cleavage by the protease, because the genome, the protease is bound to the genome, so it cleaves also. And we've been looking at the structures of these genome-less proteins, and I will just show you what happens at the vertex region. We have proteins 8 and 3A in the cleaved state, and now you can see how the vertex changes. And one of the changes that we see is that when protein 8 matures, it is kind of contracted and it stretches out and pushes this yellow domain here from protein 3A from its location. So we no longer see it because now it's not ordered. And we also have this small thingy here that changes. And this small thingy turns out to be also part of protein 8. This is the change for protein 8 during maturation. This is before maturation. This is a single polypeptide. After maturation, it is cleaved, and the cleaved peptide is down here, this L shape. And you can see here the conformational change. Here we have an alpha helix, now we have a beta sheet after the cleavage. So this protein is like a coiled spring that during maturation just stretches out, changing all the interactions here in the vertex region. So, just to take home messages from this talk, we have seen that the adenovirus core proteins do many things apart from just helping to squeeze the DNA inside the capsid. They regulate penton loss, they regulate genome release, and the exposure of the lytic peptide during entry. So they are just not, as I said, pushing the genome inside the capsid. And then for the minor core proteins, we have seen that they are plastic. They change depending on the context, depending on the virus, or depending on the maturation stage. So, this is it. These are the people who actually did the work. In red, the people involved in the AFM studies, and the funders, and I apologize for being over time.